All right, we'll start off on time. Hi, everyone. So I'll just start everyone by acknowledging the Boonwurrung and the Wurundjeri clans of the Kulin Nation, who are the custodians of the lands and waters, and pay our respects to elders past, present, and emerging. It's my pleasure today to welcome you all, those online. It's a short intro from me. I'm just going to tell you that there's lunch outside afterwards, and those online, if you're in your offices, to come down and join us. Is that right, Terry? I think I've remembered all the words. But it's a great opportunity today to hear from a recruit from a year ago, but I don't have the opportunity to introduce him. I'm going to pass it across to Mark, who's been kind enough to leave his very flashy office in the new Paula Fox building, which is glary but stunning, to join us plebs over here in the old Alfred Centre. So,
thanks very much, Ben. Yeah, I just thought I'd slum it for an hour or so over here in the Alfred Centre. No, it's really it's a great pleasure today to introduce Associate Professor Vivek Naranbhai as our presenter for today's seminar. Vivek's actually been on the campus more or less for close to 18 months and is which is the result of a very, very canny recruiting strategy by Professor O'Brien and Professor Jane, who enthusiastically, and actually Professor Pellig, I'll give Anton his credit, who very enthusiastically pursued the recruitment of Dr. Christine Chang into the infectious diseases group here. It just so happened that Christine was married with a couple of children to Vivek and essentially required him to come across also as part of that recruitment. So, and you know, we could not have been more excited about the possibility of him landing within our precinct, and everything that he's done and is doing since he's landed here is consistent with that level of excitement. So Vivek's CV is really off the charts. If any of you think you've got a good CV, you should have a look at his. So he is an oncologist with a particular interest in thoracic oncology, medically trained in Durban in South Africa, and eventually was on the senior staff at Massachusetts General Hospital in Boston. And in fact was the chief resident, I think, there. Is that right, Vivek? Which is quite an honour in itself, and reflective of the esteem with which his peers, at least there, held him in. Academically, he's had a really spectacular trajectory, including a stint in Oxford University as a Rhodes Scholar, essentially en route to Boston. But you know, at every stage of his academic career, has really achieved spectacular things, and we might hear a little bit about some of those today. He is currently in the process of being recruited and supported to recommence his clinical work at the Alfred, but has already, from the university's point of view, got his own group going, bringing in funding already, and starting to generate some really interesting and exciting projects that I'm sure he'll tell us more about. So Vivek, formally in front of an audience, welcome to the precinct. It feels like a long time since you've been here, and we really look forward to hearing about your work today. Thanks.
Thanks so much. Thanks so much. Thank you very much, Mark, Terry, Steve, Jay, and Herschel, many others who both recruited me, but also have made me feel comfortable and at home here. And I'm glad to see many of my friends from the ACBD. I think pretty much every lab head in the ACBD has welcomed me, and we've got some sort of collaboration going. In the course of the next 45 minutes, I want to share with you a little bit of what our lab's doing. And I've got several threads to this narrative. The first is that I will unashamedly try to tell you about myself and my group, because I'm hopeful that some of you may want to work with us. The second is that I want to tell you a little bit about our tremendous passion for the proteasome, including this little fellow who's the proteasome penguin and is our lab mascot, and comes along to our talks and was knitted by one of our medical students in our lab, Joyce. So I want to share with you our passion for the proteasome and T-cell receptors. And the third is to perhaps share with you then where we think we're going to go. And we have rather ambitious goals for ourselves. We may fail, but I will, in this audience, share them so that you can hold us to those goals. So I've titled this talk, A Blueprint for Discovering Life's Rules of Protein Degradation and Antigen Recognition. And why did I use the word blueprint? So I don't know how many of you know, but this is the history of the word blueprint. So on the left is a cyanotype from William Herschel. He was the son of John Herschel, who sort of gave us the Milky Way, the planet Uranus and such. William Herschel was himself a tremendous polymath. He invented cyanotypes, and cyanotypes were used for photography, but also were the precursor and where the term blueprint comes from. Because by using the chemistry of ferric and ferrous ions and cyanide, you could basically create prints that you could create multiple copies of, send them to all parts of the empire, and now you could make destroyer ships wherever you wanted to make them. We're not interested in doing that, but the analogy is true in that I think that this century will see us become, rather than clinicians and pure scientists, we're going to become engineers of the human cell. And so I think blueprint is a fitting analogy. My particular interest is rubbish trucks, in particular the rubbish trucks of the cell, the proteasomes. This is from a patent for the first rubbish truck, and T cells. So on the right is a T cell interacting with the tumor cell. So as we go through this journey, what I want to share with you is really the skeleton of what we're doing. I'm not going to probably be able to share with you any conclusive story. Indeed, in a year, we've been lucky to even get a single Western blot to run straight. But I think we have a tremendous number of great people in our lab. And what I want to share with you is a skeleton of where we're going and why we think this is a really important set of questions to answer. And really, there are two motivations. The first is to discover the beauty of biology, so science for the sake of science. And the second is hopefully to do that for the benefit of patients. And that's why the lab's called the Laboratory of Translational Immunology. So what we're fundamentally interested in is how non-self is recognized and how we can use it to improve immunity to cancers and viruses. And so the lab is divided into really two focal areas and a few other sort of legacy projects. And that is the biology of the proteasome and antibody and T cell receptor engineering. We have a couple of legacy projects I flagged on the right, just so that if you're interested and you want to collaborate, happy to chat about them. For example, we have interests in FOXP4 genetic variation and how transcriptional control of FOXP4 affects risk of long COVID and lung cancer. It's a very interesting variant that causes or is linked to both. It's the only genome-wide association with long COVID, whatever that disease is. We have a NIH-U54 on COVID and HIV. It's really centered around spatial and epidemiological stuff. And then I was the founder of the International Tuberculosis Host Genetics Consortium. So whenever in recent meetings, I've gone to people who have assumed I'm going to talk about TB, but I no longer work on TB. And so the consortium has been handed over, and there are other people I can refer you to, but we have the world's largest host genetic studies of tuberculosis, now published in eLife and follow-up papers coming. And then finally, we continue to be interested in natural killer cells as they apply to viral disease and infectious diseases. And this is work that I do in collaboration with Andrew Brooks across the other side of town. And our lab uses largely genomic and immunological tools and increasingly synthetic biology and protein engineering to answer questions about the biology of the proteasome, the biology of the T cell receptors, and how we may use them for therapeutic benefit. So here's a brief picture of all the folks in the lab that whose work I'll mention in the course of today's presentation. And as you can see, it's roughly divided into folks working on proteasomes, folks working on TCR and antibodies. Some of us still work on viral diseases and antibody engineering, which crosses over with the others. And then underpinning it all is a fair amount of work on host genetics. I won't speak at all today about our work on host genetics. And you'll see there's some holes in the slide, and I'll fill those in as we go along. But these are some of the characters that you'll see participate in the story today. So as most of you will know, central to the recognition of viruses or cancerous cells is recognition of non-self. And that involves a virus or tumor antigen being chopped up by this multi-catalytic machine in the cell called the proteasome. And it gets chopped up into antigenic peptides that can be loaded with the help of chaperones, ERP57, TAP, tapacin, etc., onto MHC class I molecules, which then make it to the cell surface and are stabilized by beta-2 microglobulin and recognized by the T cell receptor. Incidentally, it's the 40th anniversary of the cloning of the T cell receptor by Tak Mak and Mark Davis this year. So it's rather apt, I think, that we're sort of focused on this for the rest of today's talk. There is, of course, a second pathway and other pathways by which antigens can be presented, including exogenous antigens entering the endosome and escaping to be presented by MHC class II. Now, for a long time, we've been interested in, or I've been interested in variation at the HLA locus and how that affects immune checkpoint inhibitor outcomes for solid tumors or virus outcomes, mostly in the case of HIV, have been our models. And these are some of the citations of work that we've done. And largely our work has involved doing genetic epidemiology and then backing it up with mechanistic understandings of how it works. So for example, HLA-A variation in HIV disease is linked by HLA-A stabilizing E and such, and that's described in one of these papers. I won't talk about that today. We've also been interested in how host genetic variation affects risk of adverse effects to therapy. And in particular, we've had some recent interest in IL-7 variants, and in collaboration with Miles Andrews and Sharon Bala, we're continuing to be interested in that. Again, I won't speak about that particularly today. But what I will speak about is how we became interested in the proteasome. And it's a story about how expression levels in the proteasome are associated with response to immune checkpoint inhibitor, starting in non-small cell lung cancer, but increasingly we're seeing it in other settings. And suffice to say that what I'll be telling you is that the proteasome system is not as simple as I initially thought. Perhaps you appreciate it. I know people like David Tarleton probably know more about these things from his myeloma models than many others, but the proteasome system is an incredibly diverse system that's geared towards protein degradation. And what I'll try to tell you in the course of today's presentation is that that's of central importance to antigen presentation, but also of great relevance to things like neurodegenerative disease and important phenotypes like aging. So in general, we were motivated a few years back by the observation that immune checkpoint inhibitor obviously had changed therapeutic outcomes for many patients. And for example, here is a graph from Bobby Ochoa and from the NCI in which they describe the association between number of coding mutations or neoepitopes and response rates. And what we observed was that, and what many have noticed, is that yes, response rates are great, but in general, they end up being sort of in the middling 40 to 60% for tumors like non-small cell lung cancer. And perhaps for Merkel cell or such, you get higher response rates. And we don't quite know why that is. And many others have been motivated by the same observation that we don't quite know how to predict why some patients respond well and others don't. Of course, there's many factors, microbiome changes, cellular factors, etc. But what we know is that some fraction of patients get a durable benefit and have prolonged overall survival. And those are the patients we continue to obviously dose with the hope of sort of trying to find. And we were motivated by trying to find out how we can predict them. Of course, these patients don't only get responses, but many of them get immune-related adverse events, inflammatory conditions of various organs like the heart, the brain, etc., can be life-threatening. And we don't know how to predict those. And again, I won't speak about that, but we have an interest in that. So with that motivation, in collaboration with a group of other centers led by Justin Gaynor, my clinical mentor at Mass General, a consortium was created to look at pre-treatment non-small cell lung cancer biopsies and normal tissue and associate changes in the pre-treatment tissue with response to immune checkpoint inhibitors. So we have either overall response to checkpoint inhibitors or in some case progression-free survival, overall survival. And what we have is whole exome sequencing from the tumor, so that gets somatic mutations that the person wasn't born with but developed over time, and are actually that drive the cancer. And we have RNA-seq from the tumor, but this is bulk RNA-seq. So what we found when we were looking at these data, and I sort of joined the project towards the second third of the project, and became very interested in a particular part of it, which was the transcriptomic findings, was that when you looked at the transcriptomic changes in association with response, I'll pop these proteasome genes. So let me walk you through this. On the graph on the left is a volcano plot showing the level of statistical significance or level of statistical association of a given gene's expression with a response. And on the right of the zero is a good response, partial or complete response. And on the left of the zero is stable disease, so tumor didn't grow, or progressive disease, so not a very good response. And what you'll see is this is these PSM genes. Now, to me, these were very exciting, because these are the genes that encode the proteasome and its associated regulators. And so when I saw these, the obvious question was, well, are these proteasome-related genes doing something to enhance antigen presentation? Or is this purely confounded by interferon gamma? Because you could say, well, you see these other interferon gamma-related genes like CXCL9 and 10, et cetera, and maybe more gamma just makes you a hotter tumor and then you do better. Got nothing to do with the proteasome. And it continues to be a central issue. How do we know this is a causal association or not? And I'll show you how we try to answer that, or how the lab's trying to get at it. But what you see, at least when you look at the transcriptomic data, is that amongst all interferon gamma targets, you see a pretty broad distribution of association statistics with response. Similarly, if you take all proteasome subunits, you see a pretty broad distribution. And in both cases, it's the genes that are driving the association are these specific immunoproteasome genes. PSMB 8, 9, and 10, which encode for the immunoproteasome, and PSME 1 and 2, which encode for this cap that sits on the proteasome. Now, some of you may be looking cross-eyed at this point, because I was when I first looked at this, because I really didn't have much clue as to what these things were doing. And it actually is very interesting, because we don't know enough about their biology, and it's why our lab is really dedicated to their understanding. So let me take a step back and ask the question, well, how do cells get rid of waste? So throughout through the 1950s onwards, the prevailing thought was that, well, most waste must be degraded by the lysosome or by the endosome. And Christian Duve won the Nobel Prize in '76 for that. More recently, we know that processes like micro and macro autophagy contribute to degradation of proteins, and Yoshinori Yosumi won the Nobel Prize in 2016 for it. But the predominant mechanism by which proteins are degraded in the cell is the ubiquitin proteasome system. And that was a series of discoveries for which Rose, Chichinova, and Hershko shared the Nobel Prize, but for which I think Alfred Goldberg was probably an equal contender. And in the original Hershko, Chichinova, Rose observations, Chichinova and Hershko had gone from Israel to Rose's lab in the US for a sabbatical. And that was where I think a lot of this was nutted out. And what they had observed was that a protein in association with some factor and in the presence of ATP could be degraded by something to develop amino acids. We now know that what happens is E1, 2, and 3 ubiquitin ligases add ubiquitin to lysines on proteins, and this targets them for proteasomal degradation. And the proteasome was that name was actually given by Alfred Goldberg. Here's Alfred Goldberg on a yacht off the coast of Boston with his "Life is Degrading" shirt. Sadly, he's late now, but the ancestry of ideas still traces a lot of what we know about proteasomes to Goldberg and his protégés, most of whom are now sitting at the University of Tokyo. Suffice to say that what the world really, when you think of protein degradation, you think of ubiquitinated proteins, which are degraded by the proteasome in an ATP-dependent manner. However, there are a number of specialized proteasomes that divorce protein degradation from ATP dependence, ubiquitin dependence, and change what the targets are that can be degraded, and furthermore, change the type of degradation products. We know this because you can, for example, observe that when you take away the normal proteasome and only put in these specialized forms, you can get totally different neoepitopes produced and presented at the cell surface. And in some cases, these are immunogenic. So let me tell you a little bit about these specialized rubbish truck bins, basically. Basically, the cell has developed different ways of processing rubbish for different purposes. So the constitutive proteasome is that very well understood barrel-shaped structure consisting of 4 heptameric rings. So there are 7 subunits making the alpha rings on the 2 outer sides, and 7 subunits making these inner beta rings. And the business end of this multicatalytic protein is the B1, 2, and 5 catalytic proteins. So these are N-terminal threonine proteases, which degrade proteins when they're fed through the barrel. And for the most part, we think that target recognition is driven by a lid particle, which I'll show you in a second. But it turns out that there are other forms of the proteasome. So under interferon stimulation, oxidative stress, ER stress, and a number of other stresses in humans, but also in other mammals and actually down to other primitive organisms, they're specialized forms of proteasomes. And in humans, we call this the immunoproteasome. And it's characterized by the change of these catalytic subunits from 1, 2, and 5 to subunits, B1I, 2I, and 5I. Furthermore, in the thymus, specifically in cortical thymic epithelial cells only, the only cell type in the human body, there's a further specialized form which takes the immunoproteasome, drops the B5I, and puts in a B5T. What exactly that does is unclear, but we know that if you delete B5T, you lose CD8 T cell populations and repertoire. So it's probably got something to do with selection in the thymus. And then there's a 4th type of proteasome about which there are about half a dozen papers published, which basically say if you knock it out, mouse don't breed, full stop. We don't know what they do. They probably do something to histone proteins. And these are characterized by a normal proteasome, but instead of alpha-4, they swap an alpha-4S, a spermatoproteasome specific alpha-4. Now, nature doesn't do things for the heck of it, right? This intricate system must have evolved for reasons. And the driving reason is that having these different subunits allows diversification of targets and products that you can produce. It gets much more complicated, though. There are a number of regulators which sit on 1 or 2 sides of the proteasome. And the classic subunit is the PA700 subunit, which is this massive particle that has ATP degrading domains, ubiquitin recognizing domains. It forces the lid open, it unthreads the protein and feeds it in. Very well studied. This is where you want to get your protein to if you're making a targeted protein degrader or a PROTAC. Some of you have them, I think, in therapeutic trials in the cancer center right now. But there are other forms of the proteasome regulators as well. And we're particularly interested in PA28 alpha beta. And that's because this is the subunit encoded by PSME1 and 2, those genes right at the top of the volcano plot I showed you in our cancer data set. There are other subunits as well, PA28 gamma, PA200, which is probably inhibitory, and PI31, which goes into the proteasome and jams it. And what exactly these things do is a little bit enigmatic. What cells express them, under what conditions, and why is a pretty sparsely populated literature. And that's where we're trying to really, I think, try to go after new biology. So these are some of the questions that we've written to ourselves in this blueprint, which I hope hurts your eyes, because that was the purpose here. So the first question is, well, should we even really study this? Is this relevant to cancer, or was that a phony finding due to interferon gamma levels, and we're wasting a bunch of academic time studying something that isn't relevant? So I'll try to convince you first that I think we probably should, and the best evidence I can come up with is what I'll show you in a few minutes, courtesy of some of our groups. And then we'll sequentially walk through some other questions about what the expression pattern is, what their subcellular localization are, how these different proteasomes are regulated, and what these combinations do. And obviously, this is all motivated by a desire to find disease applications in which therapeutic manipulation would bring about some benefit. So what I'm going to do in the next few minutes is share with you some early data we have, nothing conclusive, but early data we have trying to answer some of these questions. So first question, should we really study this? All right, you showed us data about objective response. Who cares about objective response? Do higher expression levels even matter to patients in terms of clinically meaningful outcomes? And so if you look at PSME1 and 2, these PA28 alpha beta encoding or the immunoproteasome encoding subunit, what you see is that higher expression is associated with longer progression-free survival for both PA28 alpha beta and the immunoproteasome, but not the constitutive proteasome. And a similar thing is seen when we look at overall survival. This is in about 150 non-small cell lung cancer patients treated across 7 centers. We have additional replication data, not quite as mature, that looks like it's probably going to go in the same direction, but these are unpublished data at the moment. But what this does is it advances then a testable hypothesis of what may be happening, that perhaps the higher immunoproteasome expression results in better HLA class 1 peptides, because we know the immunoproteasome tends to cleave better at hydrophobic residues, and the peptides that are resultant bind MHC class 1 with higher affinity. And maybe this drives better CD8 T cell recognition and therefore better outcomes. All right, now it's very easy to come up with a mechanistic hypothesis to back up your claim after you already have the clinical observation, but how do we know this is true? So the first thing is that if you ignore the mechanistic hypothesis for a moment and you just say, well, do you see this in any other disease? And the answer is yes, you do. You see that higher expression levels of immunoproteasome are associated with better responses to ICI in melanoma and better TIL responses in melanoma as well. And there's a prevailing thought that actually PA28 alpha beta may be marking hot tumors and PA28 gamma marks cold tumors, or PA200 rather. All right, so we see it in other diseases, but that doesn't mean anything. It could just be the same things going on. Higher interferon, more proteasomes, and that's why you see the observation, but may not be true. So how do we know if this is true? Well, one additional thing we can look at is that we can look at the TCR repertoire. So we've sequenced the TCR repertoires in those same patients that I've shown you already, and higher expression is associated with a larger TCR clonal repertoire, but not as a controlled BCR repertoire. So that's on target. It sort of is supportive of the mechanistic hypothesis. And then our colleagues in Israel published this paper saying that PSME4, not PSME1 and 2, PSME4 inhibits the proteasome, reduces peptide repertoire, and associates with worse response to ICI. So it's really a negative phenocopy of what our observation was. And I think it's very compelling data that says that actually you've got multiple parts of the system where if you have more of antigen presentation, it's better, and if you have less, it's worse. And you're seeing it in 2 independent systems with different levers. So I think that's supportive. But how else could you confirm this? Well, in today's age, we know that genetic validation of a target vastly improves likelihood of target translation into the clinic. It's sort of on the order of 2 to 4. And conversely, in Nature Genetics last week, you'll see that if you don't have genetic validation, it vastly reduces the odds of your drug candidate ever being of clinical benefit. So the way you generate this sort of evidence is obviously through multiple different ways, but in particular, in the case of these sorts of not rare diseases, you can use Mendelian randomization. So the idea here is that if a genetic variant increases immunoproteasome expression or processivity, well then, if your hypothesis is true, it should proportionately increase the outcome that you're interested in this case, outcomes from immune checkpoint inhibitor. And so that's the hypothesis you test. You have to fulfill a bunch of sort of obviously assumptions for the genetic variants to be valid. I'm not going to show you all of that today, but suffice to say that we've tried to do this, and I'll show you some of those data. So Ben Peng in our lab, in collaboration with Sasha Gusev at the Dana-Farber, my erstwhile home, has done the first steps of an MR analysis. And so what we have here is we found valid genetic variants for the immunoproteasome and PA28 alpha beta subunits. And we asked the question that, well, is a genetic variant that increases expression associated with longer survival or not? And this is very prelim data because we're using summary statistics here, not individual level data, but we have the individual level data and it's still under analysis. But what we find is that regardless of the MR method you use, you see that it's likely that higher expression is associated with better outcomes in a seemingly linear fashion. There are a bunch of sensitivity analysis you do to confirm that this is likely true, including leave one out. And it seems to support the hypothesis that there's likely to be a causal effect between more immunoproteasome and better ICI outcomes. But there are additional controls you can run. And so we ran a few other controls, and that is, we don't see any such association between genes that are part of the standard proteasome, which you don't think should participate in antigen presentation and survival. When we use the PSME4, the negative regulator, we see the opposite effect. So that's giving you the phenocopy. And if you look at patients who don't get checkpoint inhibitor but have non-small cell lung cancer in the entire Dana-Farber data set, we don't see any association between gene expression levels that are tagged by genetic variant and OS. So it looks like the controls are supportive as well. And at least the primary observation replicates in melanoma. We don't have a cohort of non-ICI treated melanoma, and we don't have adequate PSME4 data in the melanoma cohort. So this looks suggestive, but needs further analysis. But I think is likely to encourage us in terms of thinking that we should actually bother studying this. So should we really study this? I think yes. So the next question is then, well, hold on a minute. PSME1 and 2, if you go to the immunology textbook, is induced by interferon gamma. So that means it should be there in immune cells. What are you talking about lung cancer? What's got to do with lungs? And so we've had to go back and actually look at what's the normal expression pattern of this. And to do this, we collaborated with Jay Rajagopal, who's a stem cell biologist at Harvard Medical School and a former mentor of mine. And he's the guy who incidentally described the ionocyte, the CFTR expressing cell type. But what we have there is then single cell RNA-seq data from 10 individuals, adults and children, mostly healthy. One died of a pulmonary-related disease, the other didn't. And what we have is about 380,000 cells. So it's the biggest single-cell human lung data set that exists currently. Looking at that, you can use the RNA expression to figure out what cell types you have, and then ask the question, well, where is PSME1 and PSME2 expressed? And have control genes for all of the cell type-specific markers. So for example, you can use PCAM1 to mark the endothelial cells, CFTR to mark the ionocytes and so forth. And what you see is that PSME1 and 2 seem to be actually pretty robustly expressed, both in terms of the proportion of cells expressing and the mean expression in primary lung parenchymal cells. So here you've got mesothelial cells, which almost 100% of which express PSME1 and 2 and express it at very high levels. And it sort of makes sense. Mesothelial cells are sitting at the interface to the outside world. You probably want to have a way of processing antigens in an efficient way. At least that's the way we think this is. But the key point is that there is constitutive expression of these in the lung. And this probably has then disease relevance. In contrast, you don't see very high expression in the immune cells. So what we think is happening in the tumor models is that patients probably have higher PSME1 and 2 expression in their tumor cells, driving better antigen presentation. And you don't need it in the T cells or the B cells, you have it in the tumor cells. But again, this is bulk, we went from bulk RNA-seq now to single cell RNA-seq, and this is work that Christy Ying, a very talented lead postdoc in our lab, and Charlie Fung generated together. Now, what we know is that you can also find PSME1 expression in pretty much every lung cell line that we've looked at, so it's constitutively expressed. You don't have to dose with interferon gamma. And I'll show you a bit later that actually we think there's a bit of a red herring with interferon gamma. Do you see this actually in primary lung tissue? Well, if you look in collaboration with Katrina McLean and Dan Guff, at PSME1 expression here in red, compared with TTF1, the lung epithelial marker, and DAPI just for cell nuclei, or CD3 for T cell. There's plenty of PSME1 expressed in the normal lung. And if you look in multiple different regions, it's pretty avidly expressed in most parts of the lung. And there isn't that much co-expression with CD3. Now, these are obviously preliminary data. We'll have to go back and mark for stain for additional markers. And the hope is that we can then start staining tissue from patients with lung cancer. But it does look like you're seeing it at the bulk RNA level. It's there constitutively in the lung at the RNA and the protein level. So what regulates it? Now, as I told you, the classical teaching is that interferon gamma regulates it. So why would it be constitutively expressed in the lung? Right? There has to be some other way that this is going on. It's not like your lung is bathed in interferon levels, certainly not the normal one. And what we find is that, or what we think is going on, is that there's probably actually context dependence of expression. So if this has taken us a few months to get at, but when you go all the way back to original data in the 1990s, all the data that says that PSME1 and 2 are induced by interferon gamma come from HeLa cell lines. And so we've got HeLa cell lines and we've dosed it with interferon gamma, and you can probably convince yourself that maybe there's a bit of increase in PSME1 expression. Certainly if you stain for PSMB8, the immunoproteasome specific subunit, as is very well described, you can induce, you go from no immunoproteasome and you swap almost all the proteasome to immunoproteasome when you dose with interferon gamma. But when you dose other lung epithelial cells like H441 and 1573, shared with us by Dan Guff, we don't actually see any apparent increase in PSME1 expression. Maybe you can convince yourself there's a little bit by H441, but when we've repeated this, we're not able to see that it's replicable or it's significant. And the same is true for PSME2. So it's very likely, and indeed there's mouse data that's hidden in some papers going back, that interferon gamma is probably not required for constitutive expression of PA28 alpha and beta. And so that has two implications. One is that it gets rid of the issue of whether that original finding was likely to be confounded by interferon gamma. And two, it tells us that there's something else regulating it. Now, what it is, I don't know yet. We're going to try and figure it out. We have some clues in terms of what sort of stress you can induce on the cell to induce it. But it's very likely that there's some other pathway that's relevant here. And we think that if you could understand that, then you could use other genetic instruments to validate the system. So this is work done by Charles Ma and Christy Ying. Charles is a 3990 student in our lab, who, as you can see, has got some of the best Western blot hands, certainly much steadier than mine. So very nice work. Now, the next question then is, okay, well, PSME1 and 2, not interferon gamma induced, they're in the lung. Where in the cell is it, right? We don't actually know this that well, and at least not in human cells. And what we know is that these proteasome subunits are exquisitely sensitive to different stimuli. So for example, if you look at the classic immunoproteasome subunit B5I and you dose with interferon gamma, it looks like my label is in white and disappeared. What you see is a nuclear shift with stimulation in human cells. This is in 8549 cells. And so what Christy Ying is doing in collaboration with James Wistock, Geo Graham, and Ruby Law down in Clayton is trying to apply cryo-electron tomography tools to try and understand where these are and what their capping status is. Now, you could say, well, why are you using such a high-tech tool? Isn't there any other way of doing it? And the short answer is we don't think so. We've tried other things, thought very hard about it. And we know from Wolfgang Baumeister, who is not only one of the originators of cryo-ET, but also had this beautiful paper in 2015, showing that by cryo-ET, you can resolve proteasomes in a cell. This is in yeast, not in mammalian cells. And you can not only resolve that, you can figure out their capping status. Do they have one cap? Do they have two caps? Are they processing or are they sitting in their ground state? And what's the different sort of population? So you can essentially perform a whole cell census of the different proteasomes. And we think that's probably the only way you can do this, because you've got 100, exactly 100 combinations you can imagine. You've got 4 adapters on one end, 4 on the other, or 5 adapters on either end and 4 in the middle. So it's 5 squared times 4. It's 100 combinations. You can't reasonably do that by FRET or by single molecule imaging. And so we're very excited about the cryo-ET. And Christy's made some very important strides there, trying to get mammalian cells to grow on carbon and gold grids and trying to get them plunge frozen so that you can do this. And we think it'll be a very exciting technique you can use to study these organelles, just like they've been used to study ribosomes. Now, the final question is then, all right, you can do all this cool biology. What's the disease implications? Maybe it's going to be important in non-small cell cancer, maybe melanoma, but these things are really important. And I'll remind you that viruses have evolved to manipulate proteasome degradation exquisitely. Viruses, for me, are the best example of anything on Earth that has evolved to do exactly what it needs, no more, no less. And so we know that they manipulate class 1 expression. and we're trying to screen for how they manipulate proteasomal degradation. But there's also, I think, very important data coming out of neurodegenerative disease. And I particularly put this in here because I'm hoping to encourage some collaboration with folks who know more about this than I do. And in particular, the frontotemporal dementia and ALS spectrum of disease is driven by various things, but a genetic form of which is driven by C9orf72, is really predicated, or the disease pathogenesis results to a great deal on disrupting proteasome function. So this is another cryo-electron tomogram that Baumeister and his colleagues published in Cell following their 2015 paper, in which what they did was they overexpressed poly-GA. So C9orf72 is this genetic form of ALS. And the way it works is that at C9orf72, you get these glycine-alanine-rich regions. These glycine-alanine-rich regions look exactly the same as viral glycine-alanine-rich regions. And we know that viruses like EBV manipulate or can jam up the proteasome. So sort of extending on that, what Baumeister did was he generated a cryo-ET in which he overexpressed poly-GA and was able to solve for the proteasome, which are these little greenish, I don't know what color this is, green is probably my best guess, proteasomes. And you get this huge aggregation of proteasomes around the poly-GA aggregates, which are colored in red here, and you get exclusion of ribosomes. So what's happening, and he actually has single particle imaging of the proteasome, showing that these poly-GAs go into the proteasome and they jam it up. It's like taking a wrench and throwing it into your rubbish truck. That thing can't do anything more and more. You've jammed the thing up, they all accumulate, and you're in trouble. So, important question is, well, can you overcome this by putting a different adapter on it? Can you displace it by a competitive inhibitor of the catalytic subunit binding, etc.? There's obviously major implications here. And I'm just showing you one example, but there's examples in other things. Now, not all neurodegenerative diseases follow the same pathway. If you overexpress poly-Q or Huntington's protein, what you get is proteasomes have nothing to do with that. It's got to do more with mitochondria and other things. And then the final point I'll make is that actually, I think this has major implications in other diseases, and in particular, because proteostasis is at the center of lifespan regulation. So I think all of you will know there's billions of dollars going into trying to make old white men in the United States live longer. Not particularly exciting for us, still grappling with getting young people to survive childhood in Africa and Asia. But nonetheless, there is interesting biology to be learned. And what we know is that in general, those models tell you that if you restrict caloric intake or you slow down protein production, or if you increase protein degradation by, for example, treating with an mTOR inhibitor like rapamycin, you can increase lifespan regulation. And that's phenocopied with genetic manipulation in other models like Saccharomyces cerevisiae and C. elegans, in which if you enhance the proteasome, either processivity or open status, you can lengthen the lifespan of these organisms. So there's major implications. So my point here is, if we can make proteasomes better, not only will it probably enhance antigen presentation, but it may make you live longer, which is probably not a bad thing. It's better than it saying, well, you're going to die of myocarditis, and so you've got no therapeutic window here. So my main point of showing it to you is not because I care about living longer, but because I think it tells us there's likely to be a therapeutic window to proteasome enhancing therapies. And so do we have any steps towards that? Well, we know that if you target specific parts, it may allow desired, well, we don't know, but this is what the lab is really based on. We think that enhancing antigen presentation will be useful for checkpoint blockade and vaccines. Maybe it'll be helpful in organ-specific degenerative diseases. Most of the effort at the moment is going into making immunoproteasome-specific inhibitors for the treatment of autoimmune disease. Millennium, who made bortezomib, had an immunoproteasome-specific subunit, or pseudo-specific, which was crashed in 2006 in phase 1. But that's where most of the effort in this area is going. Now, fortuitously, in the lab that I was trained in with John Iafredi in collaboration with Liron Bar-Pelad and with Diane Yang, an exceedingly talented postdoc, much better in the lab than I was, on whose coattails I was jumping on for this project. What they did was they were interested in finding, and they were screening for double warhead cysteine-modified residue reactive molecules to try and get at ALK. So ALK-mutated lung cancer, really important problem. I was a lung cancer dude in the lab. They said, Vivek, go and help them and work on this. That was how this came to be. And so what Diane was screening for was therapies that can take ALK from a monomer, dimerize it, and maybe result in their degradation. We didn't know that. And what we came up was Stefan Harry's compound X1C9. And that's the parent compound that is now published and can be played with. But it turned out that as she was developing these data, we noticed something going on with the proteasome. And when we went ahead and immunoblotted for PSME1, it was dimerizing PSME1. She didn't want this, so they went on to derive daughter molecules which still dimerize ALK and kill ALK mutant cell lines, and don't dimerize PSME1 to as much as the parent compound. And when you put that 1C9 drug into a processivity assay in which you put a fluorescently tagged peptide and you look at how fast it degrades, what you see is that in these 3 cell lines, you get a dose response sort of increase in processivity. It's not very good. It's micromolar. You're not going to make a drug out of this, but it gives us a starting compound. And we think that actually there are other better places to start, in particular by peptide mimetics, which nature has evolved to manipulate the system. So we think that you can manipulate it. We have some starting ideas. We haven't yet started working on a project for somebody else to work on. So that's really what I wanted to share with you about proteasomes. And I'm going to shift in the last 15 minutes to talk about T-cell receptors. But what I want to leave you with is the idea that proteasomes are really important. Just like you need the rubbish collected from inside, in front of your house, you need the proteins to be degraded by the cell. And that isn't, the system is highly diverse and has evolved to allow antigen presentation, degradation of extracellular protein aggregates in the brain, manipulation by viruses. It's a very complex system. We've got to sort of piece it apart one step at a time. We're trying to go from first principles understanding where is it, what does it do, and hopefully we'll discover some new biology and maybe do something of use for patients. So that's it about T cells, about proteasomes. In the second part of the talk, I'm going to swap to talking about assuming that you can get an antigen to the cell surface. You can get the peptide folded in MHC class 1, and here it is on the cell surface. Well, how can you therapeutically manipulate that? Right? There aren't that many T-cell based therapies or TCR, peptide MHC recognizing therapies, but I think the world or the aperture is opening on that. And what I want to show you is how we're going to try to ride that wave. So here, for example, is a killer T-cell, a CD8 T-cell engaging with a cancer cell. You can see that you need cell-cell apposition, you need close contact. The T cell is able to eradicate the cancer cell by injection of perforin, granzyme, and other things. But the core of it relies on the T cell specifically and sensitively recognizing the tumor cell. And how does it do that? Right? 40 years after the cloning of the T cell receptor, we left with this enigma in that T cell receptors, on average, have pretty rubbish affinity for their peptide MHCs relative to antibodies. On average, their affinities are in the sort of 10 to 40 micromolar range. That's not very good, but yet you're able to get exquisite killing. Now, of course, it could be because of avidity and multiple T cell receptors. And the prevailing thought is that there could be other things going on, which I'll tell you about. But what we think is that if you can start taking the system apart, perhaps we have a blueprint for manipulating peptide MHC recognition. It's not just us, many others are in this game, and I'll show you the way that we're trying to sort of engage in this. Now, the key point is that if you target peptide MHC, you can unlock intracellular targets. Unlike antibodies, which look for surface proteins and such, or whole proteins, you can start to go after intracellular targets which are chopped up, presented to the MHC. So suddenly you can start potentially going after neoepitopes. And we have proof of concept that that works, and I'll show you some of those in a few seconds. The questions you have to answer, though, is, well, what epitope should you target? And then what TCR do you use to target it? And what we know is that in general, if you just take a TCR out of a patient with a 30 micromolar affinity, that's not going to cut it as a drug. You can't put that on a T cell and expect the T cell to do very well. It's low affinity. You've got to do something else. You've got to go and do some engineering of co-stimulatory signals. You've got to do maybe some affinity maturation. And I'll tell you what we're interested in particular is structure-guided affinity maturation and how we may do that. So I'll share with you, this is really now a story of computational and AI tools that we're doing. So the current, in 2024, where we're at with peptide MHC recognizing therapies is that we have one licensed TCR CD3 bispecific in the form of tebentafusp, which is a TCR, which has been affinity enhanced to have a picomolar affinity, so a millionfold higher than its parent, that recognizes GP100, a melanocyte-specific or antigen in the context of a common HLA-A0201. So this TCR binds to basically melanoma cells. And what it does is it engages via a weak affinity or moderate affinity anti-CD3 to a T cell. So basically you can co-opt any old T cell that's nearby to kill the tumor cell in a tumor-specific way. And actually tebentafusp works remarkably well. You don't see great response rates initially, but the overall survival data look encouraging, and it's licensed for the treatment of uveal melanoma, particularly rare form of uveal melanoma, one of the world experts of which is sort of sitting here and have been involved in the development. There are, though, two other licensed peptide MHC recognizing therapy, and those are tuberculin, which I'm calling peptide MHC recognizing, but basically it's an EBV-specific therapy that you take cells from a patient, expand it against EBV-reactive peptides, and then you can use it to treat EBV or prevent or treat PTLD and prevent EBV reactivation, a problem for our infectious disease and hematology colleagues here. And then there's lifileucel, very recently licensed for the treatment of melanoma, again, a TIL therapy. There are a number of TCR-T therapies, so T cells which have been engineered to have TCRs on them, which are going into the clinic right now, and some of which are looking promising. I think some of the colleagues here may have them in trials. And then very excitingly, there's a huge investment going into the development of TCR mimic antibodies. So an antibody that recognizes peptide MHC. Sounds easy, very hard to develop because antibodies weren't evolved to have such exquisite sensitivity. So our question is really, how can we engineer TCRs with improved peptide MHC recognition? Because if you imagine that we have proof of concept from tebentafusp and these, how do you expand this to other patients? How could you imagine that in 20 years' time, you can sequence a patient's tumor, know the neoepitopes, and be able to select the right TCR therapies for that patient, really personalizing neoepitope-specific therapy? Right now, we do that by targeted or personalized vaccines, but personalized vaccines require an immune response. And if you could just make the TCR, put what you need onto it, you could get very far. So that's what we're trying to make steps towards. So how are we doing that? Well, the first question is, well, what should you go after? What epitope should you go after? And so in collaboration with my friend Gaurav Gaiha, with whom a lot of my T-cell work is based, and with whom I've published over the years, we've become very interested in the idea that perhaps you can select target antigens based on network properties of the protein. So if you imagine the protein as a 3D network, you can model each amino acid as a node and the links between them as edges. And then you can use standard network theory, which has been very well studied in small networks about how we interact socially and how we interact on the markets and all these sorts of stuff. But you can extend the maths of that to proteins. And what you find is that that actually helps you to prioritize the right parts of a protein to go after. It's true in HIV. It's true in a paper that we published in SARS-CoV-2. And as Lizzie Rossin, who's a retinal surgeon who works with us, has shown, it helps you to identify which diseases probably, or which protein mutations likely are causal in retinal disease. But it's an open question about, well, is this relevant to ordinary cancer mutations? So Yuan Li Yunfeng in my group started tackling this question as part of his master's in data science in collaboration with some faculty of IT folks. So we've had a series of engineers and computer scientists in the group this year really to try and drive this work forward. And what you find is that if you calculate the network score of tumor-associated proteins, or either oncogenes or tumor suppressors, that the higher the network score, the bigger the loss of function. So if you mutate, this is from saturating mutagenesis published data, if you mutate a highly networked residue, you screw up the protein's function. All right, not particularly surprising. We've actually shown that before, but it sort of confirms that this is true for cancer-related genes, not just some of the old bacterial enzymes which were in the original Gaiha papers. And it's also true for TP53, although it's a little bit of a red herring. We plotted here the fitness score, which is the inverse of the functional score. But basically, if you mutate highly networked residues, you lose more TP53 function. So what Joanne and I did was we looked at, well, how does this relate to various proteins? So here, for example, is P53, which forms a tetramer and is the guardian of the genome because it sits around double-stranded DNA and is involved in double-strand DNA repair and a range of other things. Some of you will know some of these residues that I've highlighted here are commonly mutated residues. R274H, for example, a very commonly mutated P53 residue, and these tend to be very highly networked residues. So what we were seeing was just at observational level that some of these higher network residues were probably relevant. And when you look across all tumor suppressor genes, all oncogenes, and you use as a control fusion genes, now just looking at 89 lung cancer related genes, because I know them well enough to manually curate, what you see is a very interesting observation that tumor suppressor genes tend to accumulate somatic mutations. These are data from TCGA matched up with our network scores. They tend to accumulate mutations in highly networked residues. If you take a step back, that makes sense. If you're a tumor suppressor gene and you want to drive tumorigenesis, you want to lose function. So you mutate the highly networked residues. In contrast, an oncogene, a gene which needs to gain function in general to drive tumorigenesis, tends to concentrate or has no real apparent pattern of mutations. And actually you see a depletion of, you barely ever see mutations in highly networked residues. They tend to concentrate in the unnetworked or lowly networked residues. You don't see anything going on with fusion of control genes. So what we think this can be used for is it can help you to say, all right, for a tumor suppressor gene, We know that it's going to target mutations in highly networked residues. Highly networked residues, we know, are also great targets for viral epitopes. And so you can start saying, these are the ones in a list of epitopes that I would prioritize. Now, of course, that's still a hypothesis. We need to sort of show that head-to-head, if you target them, you go superior. But we're trying to use these as data to extend from what we know is true in HIV, HPV, SARS-CoV-2, retinal disease, to something that's more germane to our clinical work, which is cancer. Saida Moradvandi in our group then extended this to T-cell receptors. So now this is a little bit of a jump here, okay, because it's got nothing to do with antigenicity, but the same principles that drive a protein structure for an antigen are true for a T-cell receptor, in that if a function of a protein is a network property, you can start appreciating how, if you perturb that protein by mutating a residue, how would that result in changes to the TCR function? And what we care about for TCRs is not how stable they are, surface expression. What we care about is what is their affinity. So if you take a TCR and you mutate a residue, as people have done, and we've collected all those data very painstakingly and laboriously made our massive Excel tables, you can calculate a delta G, so the change in Gibbs free energy attributable to that mutation. And that's experimentally determined. And so what you want to find is you want to try to find mutations which reduce the Gibbs free energy, in other words, the chaos in the system, and increase affinity. And so that's what the engineering target is. And what we find is that there is at least some association between the change in network score when you mutate a residue and the change in affinity. So what this says is that if you have a parent TCR, let's say an EBV reactive TCR, and Terry tells me that he's got an EBV encephalitis patient, we need a TCR off the shelf, what can we do? My first bet is that, well, we're going to need to engineer the TCR better. And the idea is that perhaps you can start saying, well, let's mutate or such that we change the network, we increase the network score, reduce the Gibbs free energy, and we're likely to improve affinity. Now, again, we don't have experimental data to back up these, but these are data that this is our model applied to experimentally determined TCRs or experimentally determined TCR affinities that others have published. What we're more excited about, though, is generalizing this. And so in collaboration with Jeff Webb, who's a phenomenal AI researcher and sort of was one of the founders of many of the mathematics of AI back in the late 80s and early 90s, and happens to sit at Monash, we've co-supervised student Vikrant Parmar. And what we tried to do was ask the question, well, can you generalize this? Can you use structure and train an attention-based neural network model to improve on peptide MHC TCR affinity? And the way you do that is, in our case, the 6-month Masters of Data Science students didn't have that much time. So we started with an off-the-shelf model that basically riffs on the original ESM models and then tunes it for antibodies. And that was published by Shan and others who we happen to know from the MIT world. And what they had shown is that you can build a model that basically says, all right, give me an antibody, predict a mutation, then ask how does that mutant versus wild type change affinity? Well, obviously what you want to try to do is try to select mutants that increase affinity. And if you apply that model to TCRs, it's flat, all right? Nothing going on. This is the predicted delta G on the x-axis, expected on the y. All right, so it doesn't work. All right, what happens if you just fine tune it on TCRs? Well, you get something that starts approximating now reality. So these are, what we did here was we let the model learn on TCRs, we validated on a second set, and then we're testing on a totally independent set of TCRs. The model's never seen these TCRs before. So we think it's a reasonably robust method of doing it, of looking. But remember, the world of TCR structures is limited to about 400 structures right now, out of the 10 billion that are floating around your blood right now. So we don't have that many structures, but we're starting to approximate, or we're starting to have models that predict efficiency really well. I'll bore you the boring computer science, boring to some, very exciting to others. But basically we can, through a method that Jeff and Vikrant came up with, which is called reverse mutation modeling, and it's a big thing in cross mutation, you can augment your learning data set. And what you can do is you can get what seems to me subtle, but actually is significant. You can get an improvement in your correlation coefficient from 0.66 to 0.68. In additional iterations of this model now, we think we'll probably get to the just under 0.8 level by actually now training on all 400 structures or more of the data, and doing another trick that Jeff came up with, which is ensemble learning, which is, he's a world expert on ensemble learning. So what we think you can then do is basically we'll have an AI algorithm that'll beat what's out there right now at improving affinity. So give me your EBV patient, we'll have an EBV TCR, we'll predict mutations to enhance its affinity, and we'll have a better, more highly high affinity TCR. Now, of course, you could say, well, what about specificity? When they put TCRs into patients, the first time they killed 2 patients from myocarditis and 1 from encephalitis, right? And that's because of specificity issues. There aren't any good ways of computationally predicting specificity right now. We're still too early to try and dabble in that, but you can basically extemporize. If you can solve for affinity to a given peptide, you can then challenge the model with all mutants of the peptide and start saying, well, how specific is my supposed mutant TCR for something? Of course, you've got to back this all up with the experimental data, and that's where our synthetic biology workflows sort of fit in, and why we've gone after this crazy machine that Herschel has helped me to buy and Mark has helped me to buy with the help of donor funds. I'm going to wrap up here with a final comment, which is just where we're going next, which is that all of this that I've told you about presupposes that TCRs recognize MHC in a very static fashion. We're not sure that's true, right? I was with Jamie Rossjohn last week, and we had this wonderful conversation where even Jamie Rossjohn, a man who's probably solved more TCRs between him and Chris Garcia than any other people in the world, are not sure whether a TCR recognizes things in a static fashion. They've solved the crystal structures. We know what they look like when they're static, but we don't know if TCRs actually engage in shape changes, allostery or something else. And there's an emerging series of data from Ellis Renhers and Chris Garcia and others saying that maybe there's these things called catch bonds, where a TCR and a peptide MHC may approach and bind, but under the catch bond model, as you apply force to them, as happens physiologically, when one cell may interact with another, there's 10 to 20 piconewtons of force on them. What happens is you get actually increase in the bond lifetime. So in the gray line is a catch bond, and the pink line is a slip bond. But what seems to be emerging is that at least some TCRs can engage in catch bonds. Can you engineer that? So that's really the question we're trying to tackle with a new master student in our group visiting from ISA Pune. And it's really predicated on this series of data from Chris Garcia, in which 2 TCRs for an HIV peptide were observed to have almost the same affinity. One's got 17, the other 4 micromoles, not much in the real world. Okay, but one is able to bind the target with much higher sensitivity, resulting in upregulation of CD69. And this is really what's going on. You've got the MHC in pink here, holding a peptide in yellow, and in comes the TCR. And what you can start thinking about is, and what they tried to do was basically said, well, can you engineer a catch bond? A bond which, when you apply force, you increase stickiness. I won't say that we're convinced by the story, because we're not. It looks like it's really empiric. It doesn't look like there's any generalizable model here. But suffice to say that when you can go in and you can start intelligently, in their case, or perhaps in our case, based on molecular dynamic simulations, you can start guessing at mutations or residues to which you can introduce mutations, in their case, A98H, resulting in a TCR 55, which goes from a rubbish TCR to one with catch bond-like physiology. So Vinayak Anupur is trying to pursue this with a range of tools, including steered MD, and specifically we'll try and basically take an EBV-specific TCR and try to see if we can make it more catchy. So what I've tried to tell you then in the TCR model is really why we're motivated to try and find better TCRs, why we want to infinity enhance them. And our approach is trying to do this by AI-based tools. Of course, the standard in the field is yeast display. We don't have 8 years to do it. That's what it took for tebentafusp. So we need better tools. And we think that right now, this is a good application of AI. So with that, I'll sort of summarize saying, we want to make our rubbish trucks into beautiful sports cars, and we want to make our TCRs into super killers. And the lab obviously is now been here for a year. If we can get our paperwork in order, we can hire the 2 additional postdocs to run the TCR-based work and the viral-based work. And I don't know if Patrick McLean is here or not. He's a newly minted DPhil from Oxford, who I've been trying to convince to come and bolster our host genetics work. He's I think intern day 2 at the Alfred, just come back from Oxford. But we're hopeful that we can sort of augment this team of very able scientists and apply them to important problems. With that, I'll thank many people who have helped us, in particular, many of the folks who are in this room who've shared reagents, often helped us out when we're like short of a library kit here or can't run a gel or whatever. But thank you all very much, and we're hopeful that we'll be able to collaborate more. Thank you.
Any questions?
What's the realistic timeline? I'm interested in your TCR cloning in order to optimize a bespoke CAR T therapy. We're putting up 2 machines for bespoke CAR T, and realistically, we just have the company products of the show that we use. So, what is the real feasibility of having patient-specific TCR cloning? And now that we can put those TCRs in CAR T, cells pretty easily in standalone models. What can we do? What's the timeline?
Yeah, it's a great question, Herschel. I think there's two issues. One is cost of goods and complexity, and so your failure rate, which I think even with CAR-T, there's a not insignificant failure rate. And the second is timelines. Can the patient really wait for you to go do your stuff in the lab and come back to give them the therapy? This is why I don't think that I would be surprised in 20 years if we're administering CAR-Ts or TCR-Ts. Live cell products are too expensive to make, too complex. We're going to get these T-cell associated leukemias. So we're placing our bet on the idea that you can engineer small molecules to have the functionality of a T-cell. And we think that maybe there you can get it down to a few weeks, totally speculating, but I'm guessing you need a few weeks if you've got a patient in front of you that needs something. Steve Rosenberg had this paper in Nature Medicine 2 weeks ago in which they take colorectal tumor, take it out, exome sequence it, let the tools come out of the tumor explant, expand the tools in an antigen-specific way, and then reinfuse. And he did that in a seemingly enough time to get it back into the patient. And he got, I think, of 5 patients who had T-cell expansion, 3 had responses, proving the idea that you can, like, the pathway is right. But it's incredibly complex. And I mean, what they publish tells you what it worked at. I don't know how many times they've tried and it hasn't worked. I know your friends up at Queensmead or Westmead, I think, are trying to do similar things. But my general, I'm a pessimist on cell therapies. I think cost of goods is, I don't know, like India can make it at under 100K a patient. The rest of us are it's going to cost us 200 to 300K. I don't really see how that would be competitive with a small molecule.
And what's the repertoire of proteome expression profiles in myeloma? You focus on obviously on non-small cell lung cancer, but myeloma is the disease.
Short answer is I don't know. I know the person who's characterized that best is Cassian Yee at MD Anderson, who I think has extensively done that. Do we really need, I mean, with the number of myeloma therapies we have, I'm sure we're going to need more therapies, but it feels to me like it's a smaller and smaller slice of the pie of patients who are going to need rescue therapies. We've got DARA, we've got all these fancy regimens. I'm not even keeping up. What I think is maybe there you can give a better proteasome inhibitor and give better maintenance therapy. And bortezomib is not the best, and I think you can probably get better proteasome inhibitors.
I'm wondering about the role of the target cell versus the DC more of the proteasome and if you're looking at the DCs.
Yeah, great question. Short answer is no, not yet. It's a hole in the story. We need to. The DCs are doing something, and what exactly it's doing is, I mean, short answer, I don't know. What I think is that, so Ken Rock, who was trained by Goldberg and is now unfortunately ailing at UMass, is the guy who knows the most about this. And I haven't been able to talk to him, but we're told, people who've spoken to him say that he thinks that DCs are like a great model because they have very specialized forms and they're shuttling it to the right part of the cell, in particular PSME1 and 2. I think Christie's mentioned this as well. Christie comes from Paul Herzog's labs, has a lot of experience with interferons. And I think at some point we need to look at the DCs and where the proteasomes are and what they're doing, but we haven't yet.
That's where the checkpoint blockade is.
Probably.
Targeting, right? Or do you think it's also targeting interaction?
Don't think I know. I think I don't know. Is it the tumor cell presenting it? Is it the DC who's capturing antigen presenting? I don't know. Great question. I'd love to work with other people who know more about DCs than we do.
No.
Okay.
Thanks so much, Vivek. I think time's up. Thank you.
Okay. I will end with one thing, which is that I have brought some donuts for us, and they're not ordinary donuts. And the reason they're not ordinary donuts is because a few months ago, Joyce, I think in our lab, brought us these proteasome donuts. So you guys can enjoy these proteasome donuts with your lunch outside. But as you do that, notice that they're not actually pure, proper proteasomes. These are another subunit that doesn't yet exist in humans. They've got 8 subunits instead of 7. Not quite right, but go and enjoy.
You never get that from a diabetes program seminar, do you? That sort of quality. So thanks so much, Vivek, for a tour de force presentation, and we look forward to following your trajectory with us on this precinct in the coming years. So wonderful seminar. Thank you. And I believe there's lunch outside.
Yeah.
That's